So we have the pleasure to have today with us Michal Minczuk. So he's originally from Poland, where he did his training in biotechnology and where he did his PhD in the University of Warsaw. And then after he moved to the UK, where he worked with Aaron Cloak at the MRC. Then after that, he joined the MRC Mitochondrial Biology Unit as an investigator, and after that he became a MRC investigator. He has a great CV. You can tell by the number of awards and fellowships. And most recently, he has co-founded the Pretzel Therapeutics, a startup biotechnology that focuses on development of therapies to treat diseases that are driven by mitochondrial dysfunction. So thank you so much for your time. So,
pleasure.
Can you tell us a little bit about your research?
Sure. So thanks very much for inviting me here. So our research has been focused on mitochondrial genome. So in addition to the DNA, the main storage of genetic information in the cell, which is the nucleus, a few genes, 37, are also encoded in the mitochondria. And surprisingly, we know very little how this genome is maintained, how this genome is expressed, mainly because we don't have methods to modify it. So my lab is very busy in devising methods to genetically modify the mitochondrial genome. And we do it for two purposes. First being, we want to be able to mutate it and see what happens in order to study genes in mitochondria, how they're regulated. And the other focus is on correcting mutations in the mitochondrial genome, because unfortunately, if we have patients with mutations in mitochondrial genome, they suffer from quite devastating diseases. So this is the key focus of my lab.
Very cool. So how did you realize that you wanted to become a scientist?
That's a good one. I think to be honest, the one of the key moments I remember is when I got a microscope for my one of the Christmas presents, and I started looking under the microscope into different elements of life, and I found it fascinating. And then with that, I went to the secondary school, which was focused on biology and chemistry, and that sort of I further developed this interest in life sciences. And then there was university when I embarked quite early on into involuntarily into a research on mitochondria in yeast, and it was probably second year, and I kept digging it into the biology, and then I kept discovering new things, and then discovering mitochondria and sort of realizing how little is known and how interesting these organelles
and important too, right?
And that's sort of shaped my career, I guess.
Very, very interesting. So we already skipped one question. So how did you decide to work in the UK? This decision influenced your research and your career?
So then a little bit of in life, quite often you have these moments where things happen a little bit randomly. So I went to do a summer studentship in Cambridge, and then and sort of became fascinated about the way how Aaron's Cloak laboratory could engineer proteins so they can now bind DNA where we want. And I started to think, how can we use that in order to manipulate genes in mitochondria, and also how to edit genes in mitochondria. And that was the only lab really at the time, in Europe at least, which would be helpful in realizing this. So I asked, can I do a postdoctoral research there? And initial intention was to go there for 3 years in 2004 2004. Now we have 2023, and I'm still there. And then so yeah, I did the preliminary research in this respect, and then things were going well. I decided to stay for another postdoctoral sort of job, and then after that, I became a tenure track group leader in Cambridge. So a little bit of a by chance, by applying for this
and things
summer
are working well,
studentship,
right?
but then, and then being sort of open to a not to set everything in stone. That I really want to come back in 3 years. That's it. Yes, to be a little bit more flexible and adjusting to the situation. And also, this is where science drove me a little bit as well. Because you do the first step and then something works. You do the second step and you don't want to give it up.
Exactly.
If it's
but also science influences your life, right? Because you're a person, you have your personal interest, your path, right? You're away from family, but being a scientist also influenced you as a person, right?
I think that's the only way you can do science, really. Part of it must become your life. Otherwise, it's not gonna work. You cannot do it just as a job. It's not gonna, in my opinion, it's not gonna work.
I think so. I think the same. So we are getting your work is basically mostly basic biology. How did you decide to start a company?
So it's to do with the methods of modifying the genome. So at some point, after testing them in cells which grow in plastic in vitro, we moved some of these technologies to in vivo, to testing them in animal models, and that yielded enough preclinical data that could actually make these tools potentially translatable into the clinics. And then and these are potential so-called biologics, meaning the advanced medicine that require quite a lot of funding in order to develop them. And in my view, it was only possible with in commercial sort of and more company settings. And this is where the idea of setting up a company, getting investors on board, and develop that into potential therapeutics. That's why. So it would be. I think it's to do with the speed. How these things can be developed. Because in academia, we can also try to provide.
No, but the part of the clinical trials, you need the money. It's a big load of money that you need. And
when it gets into the work towards a phase 1 or phase 1/2, the money which are involved into production of a delivery of biologics that can be delivered into a human being, these are enormous amounts of money. So,
exactly. You need a company and investors.
So that's what I thought, that this technology is ready for translation, but just needs quite a bit of funding. So
how do you think the basic and applied biology can coexist with your experience now that you have the parts of the coin?
I think any applied biology must have very strong foundation in basic science. So we have to understand how things work in order to make a drug out of it. Otherwise, it will be very difficult to do some random changes and see which works better. I think sort of mechanism-based therapies will always have a chance of working in the future than sort of a little bit the ones that are not, where the molecular basics are not fully understood.
Exactly. So the last question: what would you like to answer during your career? What would be the big question?
I think it's not much of a question, but I think I would like to see the tools that we developed be helping patients with mitochondrial diseases. That would be something.
So cool.
This is sort of
would be amazing, right?
That is sort of my dream.
Let's fingers crossed, and good luck.
Thank you very much. Yes.
Yeah.